Broadcasting live from Miami, Florida. Today we have another in the presentations of Richard Mendel, MD, the spinal neurosurgeon from Tampa and St. Petersburg, Florida. Today's Richard is going to be talking more about optogenetics. And we have a distinguished panel, including a new neurosurgeon. We'll start with Dr. Hamir. Dr. Hamir, could you please introduce yourself?
I'm Dr. Hamidi, a neurosurgeon. Right now I'm living in Toronto, Canada. I'm graduated 12 years ago in Iran.
Great. Very good. Welcome. I hope you participate regularly. And Frederick.
Hi guys, Frederick from Belgium here. Yeah, thrilled for another great hangout by Dr. Mendel. Will be great as always, I'm sure of it. Yeah, looking forward.
Welcome, Frederick, and we hope Frederick brings some Belgian neurosurgeons on board, and like Mohammed has done. Welcome, Mohammed.
Thanks for having me. I'm glad to be here today. My name is Mohammed, and I'm joining from Windsor, Ontario. Thank you.
Yes, welcome, Mohammed, and he aggressively recruited the new neurosurgeon, and Simon, my main man.
Hi. Hello, everyone. I'm Simon Downs. I'm coming to you from Tokyo, Japan. I'm a psychologist and medical student.
And Ben, the guy that'll do a hangout from his smartphone. Welcome, Ben. Ben, unmute yourself, Ben. Well, okay.
Maybe
well,
he's not able to.
I guess we'll go on to the presentation. Welcome, Richard. You're all set.
Sure. I think that last week some of what I presented probably wasn't as clear as it could have been. And that probably is because I had an optogenetics talk to give, and the Friday before, I think it was the 7th or the 8th, the issue of science had just come out. So what we talked about instead was the problem that neuroscience is having, because nobody knows how to classify all the different types of neurons there are by morphology, and a lot of people didn't understand how to functionally map things out at a neuronal level. So I presented a lot of that, and that was from Emily Underwood's article on the brain's identity crisis. But what I'm going to present right now, and it's going to be very short, just to clear a bunch of things up. Optogenetics, last week I talked to you about Santiago Ramon Cajal, and he was like the father of neuroscience. He was from Spain, and using microscopy, he viewed a lot of the different neurons, and then at the same time there was a doctor named Camille Golgi who used a silver nitrate stain that's now called the Golgi stain to stain the different neurons. But this is where the specificity and sensitivity comes in. Optogenetics is really specific. You can really make each neuronal group very specific by stimulating it in several different ways. So optogenetics is the combination of both optics and genetics to categorize a neuron's function. Because, you know, a lot of neurons may have similar morphology, but their genetics are different, and so they may serve completely different functions. The trouble with using electricity or like with deep brain stimulation or with ECT is that you only have one option, and that's to stimulate the neurons. And you're not stimulating a sensitive group of neurons or a specific group. You're completely turning on everything. So you fire, what you do is depolarize and fire every neuron when you're using electricity as the modus. And so like deep brain stimulation is just, you know, a microelectrode placed deep down in a collection of cells and stimulating it, but not in a very specific manner, just in the general manner So, like in something with like Parkinson's disease, the subthalamic nucleus is so far the best place to stimulate because that does seem to affect the tremor. But you know, if we knew not the molecules and necessarily, but the circuit that the neurons function in, you could interrupt the circuit in a bunch of different ways. It wouldn't have to be necessary through the subthalamic nucleus. So the question is that, and this is kind of, I think, kind of hard to conceive. So I'm not going to explain it nearly as well as some of these videos that I I categorize for you. So this is the general idea about optogenetics, okay? And if something's not clear, stop me, okay? So the first thing we talked about was sensitivity versus specificity. So electronically stimulating a neuron is not a specific way to evaluate its function because all you're doing is sending electricity through it. But the beauty of optogenetics is that you can discern the function of a neuron by genetically modifying the neuron using a virus, an encapsulated virus that has what are called rhodopsins, channel rhodopsins in them. So what happens is, like when you look at algae, remember there's different algae that actually lights up at night, right? The green algae that'll light at night. And so someone got the idea of incorporating the rhodopsin or the channel for color from the algae. These are single-celled mechanisms, and you incorporate that via something like a viral vector into the genetics of a small rodent mammal like a rat, and there's also a few bacteria that live in really high salt environments that also have rhodopsins that could be genetically kind of added in via a virus. So what you do is you choose a rhodopsin, and the rhodopsin is just a gated channel that's sensitive to light. But light here is the key. So you deliver it to the animal, and they incorporate it into their neural structure, brain, and now you've got a ton of options on how to stimulate that particular neuron. So rather than like a drug that's going to affect everything it comes in contact with via the circulation, the ability to establish circuits and connectivity is much more sensitive. So what you have then after you combine wavelength and color and the ability of these rhodopsins to differentially stimulate different populations of neurons is a really sensitive toolkit. And one of the articles last week was about increasing the power or the discrimination of the optogenetics toolkit. So the reason this becomes a lot more important is that even though Ramon Santiago Cajal is the father of neuroscience, if you remember, I told you he said that he thought the retina was the most beautiful of all the neurons in the brain and visual system, and he described 5 neurons, and that's what I learned in medical school, 5. This is the 80s. So now what we found out is, and Cajal was right, using a microscope you really could only tell 5 different cells apart. But the fact of the matter is, once you splice in the ability with a light-sensitive channel, you know your light-sensitive cells, your non-light-sensitive neurons. If they're light-sensitive, you could then stimulate them to different wavelengths, which, you know, to us is color, green, blue, red, spectrum wavelengths, and you could turn specific neurons on and off. So Cajal used a microscope and it allowed him to observe, so we had, you know, morphologic information. But Desroth and the people that have really pioneered optogenetics from about 2002 to now, they had not only the ability to use functional MRI and optogenetics, but it provided them a much more varied toolkit. So you could use any one of the permutations of red, yellow, green spectrum light, turning them on, turning them off. So you could much more clearly or specifically identify what that neuron did. And then with the use of other technologies like I talked about last week, which was, you know, coding the messenger RNA and finding the genetics of of the neuron going backwards in time to see even how it evolved. But these are the slides I want you to look at most. These are on YouTube. They're not all, you know, they're not manufactured by YouTube. These are great videos. And this one I think is the best one to start with. This is Viviana Gradaru is a graduate student in Dr. Dissaroff's lab. Dissaroff's lab at Stanford is, and Dr. Boland's lab at MIT. These are tremendous. They made tremendous breakthroughs. So she's a big deal, and she gives a TED talk, and it's at the TED Caltech lectures. Viviana Gradaru, and she calls it brain control with light and applications. So if you look at that presentation first, I think she gives a really good introduction to what she's doing and what optogenetics can do. Now, these were, in my opinion, the six videos I like the most. Okay, the first one is called Eye Biology, and it's microscopy and optogenetics, and Carl Dessaroff gives the final lecture. This is actually the final lecture of a series, and then the MIT explained optogenetics lecture is very good. Simon, I think this is the one you also sent to me.
I see.
Now, the third one, this one here, Optogenetics and Other Tools for Analyzing Engineering Neural Circuits. Visually, it's great because they just show you by varying the kind of algae or the kind of bacteria they splice in, you get all different variations of how that neuron functions. I'm sorry, went backwards. Now, Ed Boyden has a talk called A Light Switch for Neurons. I mean, these may sound simple. They're not simple. It's complicated science. You'll get a kick out of it, I think. Dessaroff, who's, you know, one of the pioneers, probably going to win the Nobel Prize for this, is called Breaking the Wall to Neuroengineering, and he also gives this lecture, the Allen Institute, which is at Google, the Allen Institute for Brain Science. That's optogenetics, or how to manipulate neurons with light. So you're visually going to learn much more in a few minutes once you see the diagrams of the different gates being spliced in. And then Viviana, who I spoke about here, Gradanaro, she shows a rat in a sink, and it's a large sink. And what you see is that she put, this is several years old now, she put a bunch of she genetically modified or stimulated the right side of the brain in the rat. So it, of course, controlled the left motor system. And so what you see is a rat with a apparatus coming out of its skull. And this is wire controlled then. And you'll see a blue light goes on, and every time she stimulates with the blue light, the rat will start running in circles because the left side of the body is being stimulated, and so the thing goes in circles. And as soon as she turns the light off, it goes back to regular behavior. So, you know, that becomes pretty interesting. But then Desarov talks about, you know, the population of the hypothalamus and all the different cells in the hypothalamus. And what he says is that, you know, morphologically, these cells all look alike. But of course, as you stimulate in different areas, you know, you have behavior characteristics of community well-being, functioning with others, thirst, hunger. So you have all these different populations, and the morphology is similar, but once you stimulate, you much more clearly categorize what circuit that particular neuron is involved in. So the last article was from Science last week was wireless. Now you have the ability to stimulate the neurons with a wireless technology. So it's just like, you know, an iPod. Now we can just switch on Wi-Fi or Bluetooth, and you could, you know, stimulate these rats' brains, you know, after they've been treated with the genome splicing or the vector that's introduced it, and you could discern a lot. It's like magic. You discern a lot of behaviors just by exposing that brain to different wavelengths of light or intensities of light. So I wanted to clear up, because what I was trying to do last week, and I think I don't think I was clear enough, was that I wanted to lead up to optogenetics, because I think that's the big breakthrough over the last decade in terms of methodologies. But you know, what I mentioned last week wasn't necessarily all optogenetics. Some of that, like the mRNA sequencing, is just a sequencing method, but it, you know, it couldn't be more sensitive, because you're finding the actual DNA code to individual animals, and what you're able to observe from that is that, you know, things like brain folding, you would think that it would be identical in twins. It's really not at all. It's not all that heritable, and brains don't fold necessarily in the same pattern, although they look the same once they're folded. So all these different technologies in neuroscience are going to lead to big advances clinically. For instance, instead of a neurosurgeon just sitting there and carefully delivering a microelectrode to the subthalamic nucleus, it may be that you can map the circuit in a much more easy area to stimulate and turn the signal off, the particular circuit off. So, you know, although, you know, it's great to determine the susceptibility of a molecule or a mechanism of action that you can treat with a drug, you're going to treat every cell that it comes in contact with. But if you're going to give somebody electroconvulsive therapy, their entire brain is going to you're going to deliver excitatory stimuli, and that's why seizures are so common with ECT. So now you'll be able to deliver light information to the brain, to the specific neurons that you know, are to respond to that, and yeah, it's going to be much more elegant than anything I'd ever thought of, I'd imagined as a medical student. And that's really about it. I think the take-home thing for you is just to look at these slides on the videos, and that's Viviana Gradinero, and then look at some of the YouTube videos, and the diagrams and the behavior of these rats is kind of amazing. But that's really it.
Perfect. Well, thank you, Dr. Richard.
Thank you very much.
All right. Is there anything that I wasn't clear enough about?
I think it's interesting.
Yeah, I'm sorry. Yeah, I'm sorry. Thanks, Mohammed. Thank you very much, Richard.
We got an echo.
Okay, I'll let you take over, Simon.
Thank you, Dr. Mendel. I just had a question. The takeaway from this, that you're leading up to the lecture of last week. It seems like this is an introduction to last week's lecture, and then jumping from that to my first question would be, stepping farther into the future, are you expecting that some elements of neurosurgery will be replaced as we come to really understand optogenetics, that we will be able to manipulate neurons in the brain, or you're talking about epilepsy or Parkinson? How far?
We're going to get more. We'll be able to deliver more specific treatments in the future. I mean, that's why technology evolves.
Yes.
Everything's going to be different.
That's exciting.
And the thing is that, in neurosurgery, the basic things of head injury and subdurals, epidurals, that doesn't go away. But the more cruder way of stimulating in functional neurosurgery isn't just to be plunging an electrode into the brain hoping you're hitting the right neurons. Now you're going to have, because one of the big problems with deep brain stimulation is a poorly placed electrode. And a poorly placed electrode doesn't mean that the surgeon was incompetent. It means there could be a few micrometers difference between that target population of cells and what is generally believed to be the nuclei controlling the problem you're trying to fix. Your margin of error is just too large, and all that's been determined on the Schottenbrand Atlas, which is only 13 brains that were cut. I think they were all Europeans too. There was no, ethnic variety in those brains. So, I think there was a high margin of error. I would hope that as we go into the future, we'll get less and less invasive and more specific.
Yes.
I mean, that's progress, right?
It's it reminds me of a scene in Star Trek where Bones was looking at a recent traumatic brain injury, and they were suggesting that they make burr holes, and he's like, "My God, man, this is from the 20th century. Recently, now we don't do it." And he takes out his little light thing and he puts it around, and he fixes the whatever the problem was. And I was like, I wonder how far we are away from that.
You know what? The Peruvians 2,000 years ago were doing burr holes, so
okay.
We're not that much further ahead of the Peruvians than the guys in 2050 are going to be compared to us now.
You mean the Incans from Peru?
Yeah, they were drilling burr holes like at least 2,000 years ago.
I see. I wonder if we could ask you to come off of screen sharing, if that's possible.
Oh, yeah.
Thank you.
Green?
Click on the green arrow. There you go.
Thank you.
Yeah.
Okay.
Dr. Mendel, I'm just wondering, I have a question about the wireless technology and optogenetics.
Yeah.
So you mentioned they introduced virus to control the neurons, or
yeah. Now, I have to admit, I haven't seen it yet. I just read about it last week when it was introduced in Science. Mm-hmm. But if you look at all, like if you watch Viviana's presentation,
Okay.
you'll see the rat in the sink running in circles when the light goes on. But now you don't have to do that. You don't have to build a catheter into the brain anymore. There's a technique to deliver it without any, or completely wirelessly.
It is just like genotherapy, you just, for certain physical disorders, like you introduce this bacteriophage into the cells, and then you try to, cure those diseases. Gene therapy.
Yeah, the thing is that there's so little real big gene therapy going on. Like there are no trials in the U.S. where genes are being introduced to humans probably because of what happened at the University of Pennsylvania in like 2001 or 2, and a young kid died of a disease that wasn't deadly, but he had an anaphylactic reaction to the stuff that was being used, and it was probably unethical because the president of the medical school then had a controlling interest in the company that was doing all this, so it was bad news. But in Europe, I think there's only 1 or 2 studies going on now, and so like you were saying about the bacteriophage or whatever the virus is, what is that?
That's what they use it as a.
Yeah, they're using like a herpes vector.
Yeah, they transmit the bacteria.
Yeah, they put the virus, the vector, into the incorporate. Yeah, they incorporate it, and supposedly it incorporates relatively easy in these particular rats in Deseroth's lab. But how they, I guess they're putting some type of grid in that can respond wirelessly and make a light source that these rhodopsins fire from. All I did was read about this. I don't know how to do this. I just watched all this. I mean, that's all.
I mean, if this goes in the future, you can control people remotely. Like, if you want something to be done for you, you can just introduce.
Yeah, it'd be great. I guess in I heard in Europe already there's several highways where you give up control of your automobile completely. It's all automatic now on certain highways. So yeah, you could send your mother-in-law off to another planet or something. I guess it'd be great.
Maybe Google is going to do that pretty soon.
Yeah, I'd like to invest in it. So.
Thank you, Dr. Mendel.
Yeah.
Dr. Mendel.
Dr. Said, do you have a question?
Oh, yes, I have a question.
Mohammed or Dr. Hamidi, you have a question?
Dr. Hamidi, you can go.
Yeah, okay, thank you, Mohammed. Thank you, Dr. Richard, for your good presentation. And I'm going to ask, this technology just target neurosensitive neurons or all type of neurons?
I think we're getting feedback.
Okay.
I'm sorry. I couldn't hear you very well.
Can you repeat the question, Dr. Hamidi?
I want to ask Dr. Richard that this technology just targeted the photosensitive neurons or all type of neurons. I think
that's an excellent question, and I would not know the answer to that normally, but I did listen to the inventor of this method, Deseroth, in a video, and we really don't know how many neurons are photosensitive, but a tremendous number of them are. And it's actually, they likened it to a computer program. They said that they didn't have to prime or do anything to certain species at all. It would respond beautifully to the bacteria or the algae that was spliced in. But they did mention that there may be differences between different species of animals, but so far it looks as though in the rat and probably in the human brain, that's all very, there are a lot of photoreceptors, and it's interesting. It's a good question too. I didn't know much about these channel rhodopsins until I started reading about them, and it's not something we do clinically. So I thought it was interesting. That's all.
Thank you so much.
Thank you. Any other questions? I mean, this is really very big.
Okay. Well. I think. Okay, Richard. I'll let Simon finish it.
Well, thank you very much, Dr. Mandel. Sorry, we have an echo here. But we've enjoyed the lecture on optogenetics. We look forward to more optogenetics in the future and more. Sorry about the echo. Thank you so much. Look forward to seeing you again.
Thank you.
Thank you.
Yeah. Thanks, Richard. Thank you very much.
Thank you.